Well, let's get started. Hello, everyone. Thanks for joining the GO-BGC-Argo webinar, which is jointly organized by the GO-BGC and the OCB program. My name is Yibin Huang from NOAA Pacific Marine Environmental Laboratory. Today, Chenny and I will monitor this session to keep it going. Chenny, do you want to say hello to everyone?
Yeah, hi everyone. I'm Channing. I'm a postdoc at Scripps, and thank you all for coming. I'm excited to hear from our awesome speakers.
Thanks, Channing. This is the second series of BGC-Argo webinar. The first webinar took place in March, which we give a broad overview about the profiling float, sensor performer, and some powerful tools for data assessing. If you are interested in that talk, you can go back to check the video record online. Today's webinar will be focused on the topic regarding the phytoplankton phenomena and ecosystem dynamics. We invite a bunch of the fantastic speakers to showcase how the BGC-Argo can advance our understanding in this field from the regional to the global scale. After that, we will open up a discussion sessions. Any question and comment are welcome. You can put in the chat or you can raise your hand, unmute yourself to directly interact with the speaker. To ensure all the speakers have the sufficient time to present their work, we will hold all the questions first and then address them in the final discussion panel. Okay, let's join into the talk session. The first speaker is Yui Takeshita from Monterey. He will give a recent update of GO-BGC program. Yui, here you go.
Thanks, Yibin, and thanks, Channing, for organizing this webinar, and thanks, OCB, for hosting this webinar as well. And so, yeah, so quickly, I'm planning to, for all future webinar series, I'm planning to start them with 5 to 10 minutes of just kind of background of what BGC-Argo is for potential newcomers, and then updates from GO-BGC and BGC-Argo, such as where floats have been deployed recently and where future floats will be deployed. So quick overview of BGC-Argo. So BGC-Argo is a component of the One Argo. So One Argo, this is this new vision was outlined in Remick et al. In 2019, and the vision for Argo over the next 10 years is to maintain a float of 4,700, maintain an array of 4,700 floats. And within this 4,700 floats, 2,500 will be the core floats, which are temperature, salinity, pressure floats. Deep floats, 1,200 of them going down to the full ocean depth, and then 1,000 BGC floats. So this 1,000, so every single BGC and deep float will contribute to the core Argo mission, or the One Argo mission, and these 1,000 BGC floats will contain other biogeochemical sensors in addition to the core sensors. And then so the rationale for this 1,000 float array size was determined through a variety of OSSI and decorrelation length scale analyses. And this rationale is outlined in this Biogeochemical Argo Science and Implementation Plan, which can be downloaded from the for the Biogeochemical Argo website. And so if you're interested in how this 1,000 float array was determined, I encourage you to take a look at this report. And so currently BGC-Argo has, float can be equipped with up to 6 parameters: oxygen, nitrate, pH, chlorophyll efflorescence, backscatter, which is a proxy for particles in the water, and downwelling irradiance. And so the goal is to maintain an array of 1,000 of these floats that have at least 5, preferably 6 of these sensors. And currently there are 468 operational biogeochemical floats. So this map is updated, I believe this is May or June. So each one of these dots is a biogeochemical float, and these lobes tell you which sensors are equipped on these floats. And then so out of the 468, 251 have more than 4 parameters on them. And then these blue circle floats are the ones that have all 6 sensors. Anyways, so in this 1,000 float array, so the GO-BGC, or the Global Ocean Biogeochemistry Array, is an NSF-funded project that we have proposed to deploy 500 biogeochemical Argo floats worldwide over the next 5 years, I guess next 4 years at this point. And so the plan of the 1,000, out of the 1,000 floats, the US is planning to deploy, maintain about half of the array, which is about the same proportion as core Argo. And then so the GO-BGC is a major contributor to the global biogeochemical Argo program and the US contribution of it. GO-BGC is in its second year now, and we currently have deployed 56 floats. So all of these yellow dots are locations that we have deployed floats over the past year. And this map can be found in our website, gobgc.org/array status. And so out of the 50, in addition to these 56 floats, we have about 68 upcoming floats that are built and ready to be and scheduled to be deployed over the next 6 months. So all of these colored points are locations where we are planning to deploy BGC floats in the next 6 months. And this map is updated daily. So as new cruises are identified and plans are solidified, we will add floats to these those locations to this map. So you can check in regularly. And in addition to, and then these floats are a combination of both GO-BGC and SOCON and the SOCON project. And in addition to the US, there's scheduled to be about 250 BGC floats deployed by our international partners. So the countries that have a committed plan to deploy floats are listed on the right. And then in general, these are the locations in the ocean that our international partners are planning to deploy floats. So you can kind of keep an eye out for your region of interest, and then so you can kind of expect floats to be deployed in these general areas over the next couple of years. Finally, I wanted to give a quick update on some of the, I think, are pretty exciting updates for GO-BGC. First, we are starting to phase in and test a new oxygen optode, the Seabird 83. So this is, which is outlined in red here. So this is a oxygen optode that can both do an air calibration and is included in the pump flow stream. So it has a fast response time and can be air calibrated. So it kind of has the best of both worlds. And so we have several of these floats deployed in the ocean right now. And then I think we have ordered 10 of them to be deployed on GO-BGC floats over the next year. In addition to this, we are starting to equip the Apex floats with OCR. So these are downwelling irradiance, 4 channels. And so we are funded to equip 20 of our upcoming floats with these OCRs. And then depending on the success, we are hoping to be able to equip all of our future floats with these downwelling radiometers. We are also in the midst of testing a multispectral fluorometer. So this is developed at Seabird. So the excitation wavelength is going to be both 470 and 440 nanometers. And then so that will give a better excitation wavelength for chlorophyll A pigments in addition to the auxiliary pigments. So hopefully this will give us a better idea about community composition, for example. And then so we just tested the prototype on a cruise, and we are scheduled to deploy, I can't remember the exact number, but probably around 10 of these over the next year. And also the group in France, Hervé's group, is also planning to deploy many of these sensors as well. And finally, the last update I want to share is that the BGC Solo 2. So this is a new class of profiling floats that BGC profiling float has become a commercial product. And so this is a drawing of the BGC Solo and a picture of one that we deployed off of Monterey Bay. And so there's about 4 of these in the water right now. And so MRV will be, I believe, will be taking orders for these BGC Solo 2 profiling floats. And with that, my component is done, so I can turn it back to Yibin and Channing.
Okay, thanks Yui for a serious, exciting update. Just a kind reminder, please feel free to put any question and comment in the chat. We can discuss and address them in the final discussion session. Now let's move to the next speaker, Nicholas Bock from the Columbia University. He will talk about the biogeographical identification of the global ocean based on the BGC flow observation.
Hello, thank you so much. Just one second, let me set up my screen here.
Yeah, it looked great.
It's all right.
Yes.
Perfect. All right. So thank you very much, Ben. Thank you very much, everyone, for joining. My name is Nicholas Bock. I'm a postdoctoral researcher with Columbia University at the Monterey Earth Observatory. And the title of this presentation is Biogeographical Classification of the Global Ocean from BGC-Argo Floats. And a lot of what I'm going to discuss today is based on results from a recent manuscript accidentally, of the same title, which was completed in collaboration with Dr. Marin Cornec at the University of Washington School of Oceanography, Dr. Hervé Claust, Villefranche Oceanography Laboratory, and Dr. Solange Dolmel at the University of Arizona. And the QR code links to the paper for easy reference in case you want to look it up. Okay, so just to give some overall context, there is, of course, a huge amount of spatiotemporal variability in biological processes within the global ocean, as captured at a broad scale here by the time lapse of Motus chlorophyll A concentration measured during 2021. And understanding or trying to model these processes at some level requires a way of partitioning the global ocean into discrete regions or provinces or biomes where this variability might be relatively constrained. And so this is sort of the goal of biogeographical classification. Many classification methods have been developed over the last 80 years or so. Maybe one of the most famous ones is the Longhurst provinces, which uses data from global climatologies and remote sensing platforms to define 56 ecological provinces within the global ocean. Other classifications have relied on ocean color data alone. So for example, in Barenfeld et al. 2005, these long-term chlorophyll A variance bins were used to guide parameter selection when modeling primary production. In all cases, though, there has been limited opportunity to account for subsurface variability in classification criteria, primarily because satellite measurements primarily reflect what is going on at the surface, while ship-based measurements have really sparse spatiotemporal resolution. Of course, the BGC-Argo network addresses both of these constraints, providing tens of thousands of profiles of biologically relevant parameters, and also providing a potential basis for a biogeographical classification that takes some of this subsurface variability into account. And so over the next few slides, I want to first introduce a method that we came up with for using a singular value decomposition, or SVD, to classify time series data from BGC-Argo floats. I want to talk about some of the biome-specific seasonality in phytoplankton distributions and carbon export that these results highlighted. And then I want to close with some further applications for the method and next steps for refining the method further. So with that, I want to take a really sharp detour to talk for a second about eigenfaces. And the eigenface concept is really commonly used to illustrate how singular value decomposition works and is pretty analogous to our approach for classifying float data. And so the idea is that you take a series of photos like the ones shown here, you compile all of this image data into a matrix, and then you perform singular value decomposition on that matrix and get a series of sort of spooky looking eigenfaces, each one representing a different component of the original or the overall variability in the original time series. SVD also provides a set of coefficients that, along with the eigenfaces, can be used to reconstruct the original images. So, for example, if you take 3 times eigenface 0, 2 times eigenface 1, 1 times eigenface 2, and 8 times eigenface 3, so on and so on and so on, you get this photo of Dwayne "The Rock" Johnson. If you use different sets of coefficients, you get an image of LeBron James or Queen Elizabeth. And so you don't just get these eigenfaces, you get this series of coefficients that provide a summary of the different sources of variability in each one of the initial images. And so in our approach to working with float data was analogous to the eigenfaces example, except instead of using these different photos, we were using 422 annual time series of chlorophyll A and backscatter measurements from a global BGC-Argo data set. And so performing SVD analysis on this data we obtained a series of eigen time series, as it were, in addition to a set of time series specific coefficients for each one of the original pairs of time series. And then performing cluster analysis on these coefficients, we were able to objectively classify our original 422 time series as belonging to one of 6 groups or biomes. This included the equatorial biome with time series concentrated around the equator, the Arabian Sea biome with time series solely located in the Arabian Sea, the permanent deep chlorophyll maximum biome with time series concentrated in the central gyres of each basin, the seasonal deep chlorophyll maximum biome with time series concentrated in the Mediterranean Sea and the subtropical frontal zone, and then the low chlorophyll bloom biome and the high chlorophyll bloom biome. And low chlorophyll bloom biomes were primarily located in the Southern Ocean, while high chlorophyll bloom biomes were primarily located in the North Atlantic. And so, because each one of these time series, each one of these points, I'm sorry, I should have mentioned, each point on this map here represents the median latitude and longitude of a given time series. And because each one of these points represents a full year worth of profile data, this approach provides a huge amount of data to study biological processes within each one of these biomes. And so, for example, calculating the average of the respective chlorophyll time series, it's possible to get a really clear picture of seasonality and phytoplankton distributions and physiology within each biome. Same can be done with backscatter time series, providing this synoptic view of particle distributions within each biome. And these plots, should have mentioned on the last one, x-axis represents month of the year, depth axis, y-axis is depth beneath the surface, and the color gradient here corresponds to backscatter values. So beyond looking at the bio-optical data, it's also possible to characterize seasonality in sort of the environmental context of each biome using float measurements of physical and biogeochemical parameters. So for example, we used temperature and salinity data to calculate the average mixed layer depth for each biome, shown by the black dashed line here. We used nitrate concentrations to define the depth and the slope of the nitrocline. When I talk about the depth of the nitrocline, I'm just referring to the depth where nitrate first reaches concentrations 1 micromolar greater than those of the surface. And we interpreted the nitrocline slope as representing the magnitude of upward diffusive flux of nitrate at the depth of the nitrocline. Then finally, we also used data from float radiometers to estimate the daily integrated part levels both within the mixed layer and at the nitrocline depth. And so these values are indirectly represented here by the 0.1% par isotherm shown in yellow. So a few takeaways from these results were that the equatorial and Arabian Sea biomes are both characterized by really high light levels at the depth of the nitrocline and nitroclines with really steep slopes. And this is reflected in part by the 0.1% par isotherm being more or less permanently situated well beneath the nitrocline in both of those examples. In both of those biomes. This is in contrast to the permanent DCM and the seasonal DCM biomes, where the 0.1% par isotherm was more or less coincident with the nitrocline, representing much lower light levels at the depth of the nitrocline. Finally, the high chlorophyll bloom and low chlorophyll bloom biomes were primarily differentiated from the others by this variability in mixed layer depth, the seasonality of mixed layer depth, and by extension, average light levels within the mixed layer over the course of the year. So this classification also provided insight into particle distributions within the mesopelagic zone for each biome, which we approximately defined as the area beneath the 0.1% par isotherm, with the 0.1% par isotherm being shown represented by this black line here, the mesopelagic zone being shown in the bracket for the high chlorophyll bloom biome. The area above the 0.1% par isotherm is defined as the productive zone. And so comparing the average backscatter values in the productive zone on the x-axis here, and comparing those values to a proxy for transfer efficiency, that is the ratio of average backscatter within the mesopelagic zone over average backscatter within the productive zone for a given profile. we were able to identify this global inverse relationship between productive zone particle stocks and export to the mesopelagic zone. I should pause for just one moment to emphasize that when we were preparing our backscatter profiles, we removed any spikes that might be interpreted as representing larger particles. So all of these results solely represent concentrations of smaller particles. But regardless, so this data also allowed us to identify that this overall trend is driven by extreme values at high latitudes during winter and summer months. Okay, so to get a clearer sense of how particle attenuation contributed to this overall trend, we also subdivided the mesopelagic zone into 4 different layers, and then calculated the average backscatter values within each one. So then we were able to compare the average particle stocks within the productive zone, shown in red, to the average particle stocks across each of these different layers in the mesopelagic zone, shown in light gray to dark gray. And the distance between these lines corresponds to the instantaneous attenuation with depth, or the attenuation in the BBP backscatter signal for an individual profile. And so highlights productive zone, highlights productive zone particle stocks to be directly related to particle attenuation, again, for a given profile with depth, especially in the seasonal DCM, the low chlorophyll bloom or the high chlorophyll bloom biomes. Okay, so that's a perfect segue to the last part of the presentation, where I want to take a minute to talk about further applications for this method and sort of next steps for building on these results. So in particular, I wanted to highlight the recent work of Elsa Simone, who is a PhD student at Villefranche Oceanography Laboratory, and who used this SVD classification approach to evaluate the functioning of the biological carbon pump in 5 biomes, high latitudes, 2 in the northern hemisphere and 3 in the southern hemisphere, as shown here. And so Elsa greatly expanded on the analyses of subsurface particle distributions for different biomes, using backscatter values to estimate particulate organic carbon concentrations, and then from that, estimating seasonal rates of POC accumulation within 8 different layers of the mesopelagic zone, as reflected by these green lines here. From this, she was additionally able to evaluate the intensity and the efficiency of the biological carbon pump for each biome, as illustrated by the bar plot on the lower right, in addition to linkages between particle fluxes and respiration rates. Okay, so in all of these results, though, the biomes remain defined as these collections of discrete time series. And so an important next step, or at least a possible next step, is to explore methods for extrapolating these classifications to areas not sampled by floats. So in other words, is it possible to give a machine learning classifier geographical coordinates and remote sensing measurements for a set of classified profiles? For example, here, the different profiles for the equatorial biome, and have that classifier predict biome membership for data that it hasn't seen before, for instance, from a satellite image. And so in sort of a first effort of this, I took roughly 26,000 classified profiles with satellite matchups for chlorophyll, backscatter, and sea surface temperature. Used them to train a random forests classifier, which then predicted biome membership in a test data set with an overall error rate of 2.5%. This confusion matrix just sort of shows the breakdown for how well it classified different biomes or profiles belonging to different biomes. And then applying this model to a global data set of the classifier predictors, it's then possible to extrapolate the original SVD classifications to a global scale, as shown here. And so while there is admittedly some sort of batty behavior going on at low latitude predictions here, a refined version of this approach would achieve the larger goal of creating a global biogeographical classification of float data, as described at the start of the presentation. Okay, so that's everything that I have. I'd like to take a quick minute to thank my collaborators in this project again, in addition to support from the NSF and the ERC funded Refine project, and of course, everyone on the BGC-Argo data management team, and BGC-Argo project in general. My email address is here. Feel free to reach out. I'm happy to share any code, data, etc. That this project produced, and look forward to whatever questions you might have. Thank you.
Okay, thanks, Nicholas, for the very fascinating talk. We will address the question in the final discussion session, but you can also directly reply to them in the chat. Right now, let's move to the next speaker, Marin Cornec from NOAA PMEL. He will discuss about the dynamic of the deep chlorophyll maxima in global ocean.
Thank you, Ibin, and thank you, Shining, for organizing this webinar, and thank you, OCB, for hosting. And yes, so my name is Marin Cornec, and the work I will present today is based on my previous PhD research, which was led at the laboratory of Villefranche under the supervision of Hervé Claustre. And so, yes, the subject of this study focused on the deep chlorophyll maximum. So what is the deep chlorophyll maxima? It's a layer of active phytoplankton at depths, which are detected from in situ measurement of chlorophyll A. So here you have an example of a vertical profile from a ship taken in the North Pacific subtropical gyre. So that show a peak of chlorophyll A, at depths. And these features, the deep chlorophyll maximum, are observed from more than 70 years ago, and mostly from in situ measurements, because they often escape to the remote sensing observation. And so there is a broad literature around this subject, but still misunderstood because of the sparse observation from the in situ measurements. So the main goal of this study was to provide a global assessment of the DCM observations, characteristic, and dynamics. And so one question that is important regarding the DCM is productivity. Are DCM productive layers? And there is like a theoretical background which shows that we have in fact a gradient between two extreme environmental systems. So on the side, we have the oligotrophic environments, which show limitation of light and nutrient at depths. And on the other extreme, the mesotrophic systems. And so in the oligotrophic environment, the limitation of light and nutrient at depths doesn't allow the phytoplanktonic cell to thrive, and then also lead to a process of photoacclimation, where the phytoplanktonic cells increase their internal content of photopigments, which lead to presence of a maximum of chlorophyll at depths, but which is not linked to an increase of biomass. And so on the extreme, in the more mesotrophic system, we suppose that we have an optimal condition of light and nutrient at depths that allow to create an optimal niche for the phytoplankton cell to thrive at depths and to accumulate. And then in this case, we have an accumulation of biomass. And so further in this presentation, I will talk about the more oligotrophic system as deep photoacclimation maximum, DAM, to differentiate them from the deep biomass maxima, DBM. And so we have to assess that those two extreme represent extreme condition, but we have to know that there is a wider range of configurations that lead to the process of apparition of deep chlorophyll maximum. So the main question from this study describe the DCM occurrence in the global ocean, their seasonality, and the main characteristic. Also regarding to the distribution of the different typology, is there deep photoacclimation maximum or deep biomass maximum, and also look at what are the main environmental drivers that lead to their presence and maintenance, and also look at how hydrodynamical features such as mesoscale eddies, which are ubiquitous in the global ocean, can impact on these features. And so for this, we use the global Biogeochemical high resolution measurements of various parameters to assess the dynamics, and this work has led to two publications around this subject. So, as you know, BGC-Argo Floats measure an array of biogeochemical parameters. And here we focus on two bio-optical parameters in particular that allow to detect and quantify the phytoplanktonic presence. So first, the fluorescence of the chlorophyll A, which is a proxy for the concentration of chlorophyll A, and then to detect the phytoplankton presence, and the backscattering coefficient, the BBP, which is the proxy for the particulate organic carbon. And in addition to these two parameters, we also have up to 4 BGC parameters that will allow to constrain the environmental context of the DCM presence. And so for this, we use the global database of BGC-Argo float profiles. So on this map, we have all the float profiles that have at least measurement of fluorescence of the chlorophyll A and the backscattering coefficients, which correspond to about 60,000 profiles sampled from 2010 to 2019. And so we first proceeded at classification of all these profiles to look at whether or not they present deep chlorophyll maximum. So here you have 2 examples of profiles for fluorescence of the chlorophyll A and BBP that does not show deep chlorophyll maximum. You can see on the first one that we have high values for both parameters in the surface, which correspond to the situation of bloom condition. And on the right, you can see that you have nearly null concentration for both parameters, which are homogeneous on the water column and which are witnessing condition of deep winter mixing, so which doesn't allow of phytoplankton in cells. And on the other side, 2 examples of DCM profiles. So you can see on both figures that we have a peak at depth of chlorophyll A, but you can see that on the left one, we don't have a peak of BBP coinciding with the peak of chlorophyll A. So in this case, we are in a case of deep photoacclimation maximum, where the cells increase their internal content of phytoplankton in response to limiting environment. And on the right, you can see that we have both a peak of chlorophyll A and a peak of BBP coincident, so which translate into a deep biomass maximum where cells are thriving and accumulated at depth. So after proceeding to this classification, we look at the distribution and the frequency of DM on the latitudinal distribution. So for this, we look at the percent of BGC profiles that presented a DCM per bands of 10 degrees of latitudes. And so we can see that around the low latitudes regions, we have an increased presence of DCM percentage, so above 50% from the equator to 30 degrees of latitude in both hemispheres. And this frequency decreases as we are going toward higher latitudes. And so then we look at the composition of this DCM profile. So to see if it's either deep photoacclimation maximum or deep biomass maximum. And what we can see is that around the equatorial water, so from 0 to about 15 degrees of latitude, the DCM profile are dominated by deep biomass maximum profiles. And conversely, in the subtropical region, so around 25 degrees of latitudes, we can see that DCM profiles are dominated by deep photoacclimation maximum. And so we then looked at global distribution of the main DCM characteristics, so the DCM depth and DCM intensity. And so for this, we computed the per band of 10 degrees of latitude, the depth of the DCM, which correspond to the vertical axis of both panels. And in color, you have on the first panel, the intensity of the chlorophyll A at the depth of the DCM. And on the below panel, the value of the BBP at the depth also of the DCM. And so we can see here also that we have like a symmetrical distribution around the equator with a covariation of BBP and chlorophyll A value along this transient. And for real latitudes, we can see that we have intermediate depths and values of the DCM. When we are going towards the subtropical latitudes, we can see that we have a deepening and a weakening of the DCM for both parameters. And finally, as we are going to high latitudes, we can see that the DCM shallows and also increase in intensity. So to resume this global observation, we have this subequatorial latitudes where DCM are dominated by deep biomass maximum, which are at intermediate depths and intensity. Subtropical latitudes which are deep, weak, and dominated by photoacclimation, and high latitude where the DCM are the most shallow and intense, but less frequent. And so then we look at the environmental drivers of the DCM with the focus on the vertical distribution of irradiance and nutrients. And for this, we focus on two main parameters. So first, the photosynthetically available radiation, the PAR, and we look at the daily dose of PAR, high PAR. And then we focus on the nitrates, which is the main limiting nutrient in the global ocean. And we look at the parameters around the nitracline, which is the gradient between the upper depleted layer of the water column against deeper re-enriched layer. And so for the daily PAR, we obtain it through bio-optical relationships, and nitrate were estimating using a neural network method. And so we look at the daily photosynthetically available radiation at the nitrocline depth, which is an index of the productivity of the DCM. And so we use it to separate the deep photoacclimation profile against the deep biomass maximum profile. And what we can see is that in the case of deep photoacclimation, we are low irradiance level at the nitrocline depth compared to what happened in the deep biomass maximum profile, which confirm the previous sparse observation and allow to generalize them at a global scale. And so finally, we look at the impact of mesoscale eddies on the deep chlorophyll maximum features, knowing that mesoscale eddies are ubiquitous in the global ocean, as shown as this map showing the sea surface height from Shelton and al. 2011. And so we can distinguish two different types of eddies according to their rotation direction from the horizontal plane. So the cyclonic eddies, which induce an upward motion of water masses in their core, and the anticyclonic eddies, which induce downward motion of the water masses, with the idea that the mesoscale eddies can perturbate the environmental condition into stratified stable system, such as the low latitude system. And so some observations have shown that those mesoscale eddies can impact on the DCM feature, either intensity or depth, but still no global assessment has been proposed at a global scale, which is the this study. So for this, it's the BGC-Argo database with the mesoscale eddy atlas, 2 eddies, which was developed by Rémi Laxonner from the ENS of Paris, and which is based on a detection from absolute dynamical topography maps. And so for each profile of the BGC-Argo database, we were able to co-locate it and to see if it was outside eddy influence or inside an eddy, and if inside an eddy, which polarity, either cyclonic or anticyclonic, and also its zonation, so either in the core of the eddy or in the periphery in the edge section. And so we focus on this collocation on the low latitude, so in the subequatorial and subtropical gyres. And we first look at the impact of the eddies, polarities, and zonation on the DCM characteristics. So we look at the impacts on the DCM depths, the BBP and the chlorophyll A at the depths of the DCM. And so we computed the anomalies of the mean properties per region. So for the profiles that were inside the different type of eddy, cyclonic, anticyclonic, core and or edge sections against the profiles that were outside of eddy influence. And what we see is that we have a global gradient between the two extreme core, cyclonic and anticyclonic, and the edge section. And we see that for cyclonic eddies, we have shallowing of the DCM along with an increase of intensity. So the increase of BBP and chlorophyll A value at its depths, and conversely in the anticyclonic eddies, so deepening of the DCM along with the weakening of its properties. And we then look at the impact of the eddies on the environmental parameters. So for this, we look at the profundeur of the depth, of the nitrocline, Z-NIT, the depth of the mixed layer depths, the MLD, and then the daily par at the nitrocline depth, so IPAR-NIT, and then the mean daily par within the mixed layer. And so we computed also mean anomalies per region. And what we observe is that we have, for cyclonic eddies, an uplift, upward motion of the nitrocline depths along with the MLD towards the surface, and so which reflects the vertical motion, and conversely in the anticyclonic eddies. And this is linked to the fact that in the anticyclonic eddies, the uplift of the nitroclines allow to have more available nutrients into more latent environments, and then which explain why into cyclonic eddies we have more favorable conditions for the phytoplankton cells at the DCM depth to thrive. And so overall, this observation in this latitudinal bands highlights the domination of the effect of the eddy pumping on the DCM properties and characteristics. And so yes, to the takeaway message is that as previously observed, the DCM are well ubiquitous feature in the global ocean, but are dominant into the stratified system, so mostly from the equator to 30 degrees of latitudes. And we show that we have three kind of different systems: high latitude with sparse but intense DCM, subtropical system with weak but mostly dominated by photoacclimation DCMs, and subequatorial regions which show intermediate values but dominated by deep biomass maximum. And knowing that cyclonic eddies enhance the biomass accumulation and potentially the deep biomass maximum, and conversely for the anticyclonic eddies. And some perspective could be to rely on the future deployment on the BGC-Argo Floats to refine this observation, and also to fill the gap within the region where we still miss some observation, and also to get into more knowing the species constituting the DCM layer to better understand their dynamics and the phytoplankton types, or for example, species succession, and also how the deep chlorophyll maximum can be quantify its role regarding to the carbon cycle. So for example, regarding the export of carbon from the DCM toward deep layer of the ocean. And yes, that's it for me. Thank you for your attention, and don't hesitate if you have further questions during the discussion session.
Okay, thank you so much, Marin, for a very great talk. Now let's move to our final speaker, Mariana B. from Monterey. She will talk a very definitely hot topic, the effect of the marine heat wave on the Northeast Pacific ecosystem. Mariana, here you go.
Do you hear me, and do you see my screen?
Yes, it's great.
Thank you, Bean. Thank you guys for inviting me to give this talk. So I'm Mari. I work at Embari as a researcher, and I work on the calibration of biogeochemical sensors from the United States side. So if you see problems in nitrate, you can blame me. So today I'm going to talk about biogeochemical floats as a framework for ocean observations. So really using the biogeochemical floats as a helper when you have additional data from other platforms. And my study case is the impact of heat waves on the Northeast Pacific ecosystem. So this is a huge effort. It's not, it's from collaboration to collaboration with a lot of other people that are here in this first slide over here. And two marine heat waves basically impacted the Northeast Pacific Ocean. One started in 2014 and finished in 2016. And this one, we have more information about it, but more recently in 2019, we had a second marine heat wave. And I am going to first talk about a little bit about the impact in organic carbon production on the first marine heat wave. And then I'm going to talk about a work in progress that we are looking on the impact of POC production and export during these two marine heat waves. And we are also interested in understanding not only the POC production and export, but if there was a change in production and export, who were the producers and who were the consumers? Did the microbial community change during these two marine heat waves, or they didn't change at all? And you're going to see that these two marine heat waves produce a very similar impact on POC production and export, but not quite what we imagined. So the marine heat waves, if we put them in the context of ocean processes, they are very broad. They can be a very small in spatial scale, or very big or a large scale process. They can also last from a few days to a few months to a few years. In the case of the Northeast Pacific Ocean, both marine heat waves had a couple of years of a time scale and in a very large time scale. So they happen all over the world. Some of them are more studied than others. And you can see that even the El Nino can be classified as a marine heat wave. In the Northeast Pacific, the ocean is very susceptible, and the phytoplankton there is very susceptible to marine heat waves, because during the winter is when the inorganic nutrients come from the deepening of the mixed layer. And this is what brings the enriched waters with nutrients, phytoplankton, to produce organic carbon in the spring and summertime when the ocean is more stratified. If we don't have this winter mixing, then we don't have enough nutrients for the phytoplankton to grow. So nitrate is an example of nutrient that comes during the winter and is available for phytoplankton, but also iron is very limiting nutrient for phytoplankton in there. So these ocean atmospheric oscillations restrict the nutrient inputs to the upper ocean. And in the Northeast Pacific, specifically, they restrict the input of nitrate and iron. So during the first marine heat wave, it was a relationship between something that happened in the atmosphere, and then there was a feedback in the ocean. So in 2014, there was an atmospheric ridge that basically limited the ocean cooling during the winter. So the ocean became warmer for that winter. A couple of months later, there was an El Nino developing in the equatorial Pacific. And this warm water from the Northeast Pacific actually had a feedback on the atmosphere in the equatorial Pacific Ocean. The El Nino intensified in the other year and actually had a teleconnection in the form of an arc pattern that crossed the equatorial Pacific. And then this arc connected the El Nino to that warm blob and made this warm blob persist for a second warm winter. So you imagine that 2 winters in a row where you have restricted input of nutrients would not be very beneficial for phytoplankton growth and for organic carbon production. In 2016, the warm blob, or the big marine heat wave, disappeared and the El Nino was fully developed. So luckily we had a bunch of biogeochemical floats there, and we could see the effect of this marine heat wave before, during, and after this first event. So this temperature, nitrate, and net community production, which is organic carbon production, which was estimated from nitrate drawdown during the summer months and then converted into carbon units. You can see that normal years, we have this well-defined seasonality. In the winter time, you have the mixing of underlying waters that bring nitrate to the surface. And then in the spring and summer months, this nitrate is drawn down and we have net community production. During the blob, this isopycnal of 25.5 was really not broken. And it's a sign that, that there was not really a very good mixing of nutrients to the surface. But in the first year of this warm event, we actually had a lot of net community production. In the second year, we had again very limited ocean mixing, and then we had very little net community production or organic carbon production. If we take a look at the Brunt-Väisälä frequency, this tells us how stratified and how stable the ocean was. And if we take a look at the halocline in that second warm winter, we see that the Brunt-Väisälä frequency was very high, which means that this halocline was just a second layer that really limited the mixing of inorganic nutrients to the surface, especially in the second year. So in that first year, then we had very high net community production, very elongated. So NCP happened all throughout the year. During the second warm year, then we had a crash of organic carbon. And then the system recovered, and we attributed this difference in NCP, even though the 2 years were warm, in the dissolved iron. So we intimated that dissolved iron limited NCP in the second warm year, and really in the first warm year, maybe the phytoplankton was highly recycling the iron that was already at surface, but then a prolonged marine heat wave would really not give a chance for phytoplankton to have any more iron available for them in the surface. Well, we didn't have iron data, and it was actually my dream to have iron data. But then this year, Taves and collaborators released this paper saying that really during the first heat wave, the Northwest Pacific was severely limited by iron in the second warm year, but not in the first. So I was a little bit wrong on the recycling iron, because what they say is that in the first year, there was a little bit of a lateral transport from the continental shelf, bringing dissolved iron. And that's why in that winter and that summer, phytoplankton was not limited by iron, and they were producing organic carbon. But in the second year, the surface was really limited by iron. There was a lack of lateral transport of iron, and that's why there was this crash in the organic carbon production. When they took a look at the community composition of phytoplankton, they saw that in 2015, there was a very big shift in the communities. So usually the area has diatoms, and in that year there was no diatoms. It was more dominated by chlorophytes, which are smaller phytoplankton. They don't take up silica, and they're part of nanoplankton. And if we take a look at the uptake of nutrients by phytoplankton, we saw that really the uptake of iron to nitrate was smaller that year, because we didn't really have iron to uptake. And then silica to nitrate uptake was negative, because of course in the other years we had diatoms uptaking. In 2015, we didn't have diatoms uptaking silica. So Traving also found out that the prokaryotic communities changed during that first marine heat wave. So they integrated the entire water column, collected DNA, and then compared before and during marine heat waves, and found out that the community composition, no blob years and yes blob years, were very different. So our question remains, what is the impact on POC production? So we would imagine that less NCP would lead to less POC production. Because the ocean is very stratified, we would increase respiration rates in the surface. So we would have less POC export. So that second marine heat wave was similar to the first one. And it was more, these warm waters propagated to the deep ocean, a little bit different from the first one. But we were really interested in seeing what was the POC. And if the POC production and export was different, who were the producers, who were the consumers? Did the microbial community change or not? So our two questions were basically, how was particulate organic carbon production and export during the two marine heat waves? And compare with non-marine heat wave years, who were the producer and consumers? So for this, we have float data in the region, temperature, salinity, backscatter, which is the proxy for POC, and chlorophyll and nitrate, which will give us an idea of how winter mixing was active during those years. And we also had phytoplankton composition, so HPLC and genetics, HPLC from Angelica, and then genetics that are still being processed. And we are also reprocessing that data for prokaryotes to include prokaryotes composition and the surface and prokaryote composition under the mixed layer depth, and maybe also include the phytoplankton composition through genetics as well. So I'm going to show you how we really need to QC BBP data. This is raw BBP collected from the North Pacific. This is the time series. So if I pick up the data from the floats right now, this is what I'm going to see. And once we QC, we really remove, again, as Marin commented, the big particles, but we also remove the background. In this background is highly due to sensor noise. And when we remove this background, we see these clean structures of BBP or POC. So what we can tell is that those warm years had a very high export of POC. This, the first marine heat wave, the high export of POC was in the second warm year. And then the next marine heat wave was immediately once the marine heat waves start getting formed. So if we separate the big particles, which are particles that are more than 100 microns, or phytoplankton aggregations, big chains, maybe zooplankton as well, we don't really see the seasonality that we see in the total BBP. So this tells us that really the seasonality was in the small particles, which makes sense because the Northeast Pacific Ocean is dominated by those nano and picoplankton. When we compare, when we zoom in those years where the POC export was super high, we can start seeing the structures. So this is chlorophyll, surface chlorophyll. The surface chlorophyll also had to be QC'd, and it needs to be QC'd. This chlorophyll was QC'd using ship-based data, but it can also be QC'd using satellite data if we don't have ship data available. And you can see that not always chlorophyll is a proxy, direct proxy for biomass. So this is BBP converted to POC. And we can see that this big export started earlier in the year rather than later in the year, and it kind of propagated. This plume of POC kind of stayed there throughout the months and really generated during the springtime in both of the years. And then we can target these areas here and these months over here and see who were producing this POC. And in fact, the floats were a little bit far apart during different years. So in 2015, our phytoplankton composition is more related to a little bit more toward the coast than in 2019. Fortunately, this phytoplankton composition was collected from the line P that crossed seasonally this area over here. So we had phytoplankton composition for both years, but just zooming in and focusing here in 2015, the phytoplankton was composed by chlorophytes and haptophytes. And in 2019, remember that this POC really started getting exported early in the year. This was also chlorophytes and haptophytes. So they are smaller size cells, less than 5 microns, and they outcompeted diatoms and dinoflagellates during both of the marine heat waves, which is very interesting because you would think that more export would mean bigger diatoms sinking because they can be ballasts, but no, it was probably chlorophytes and haptophytes. So our future work will involve targeting those regions and those years and really digging into the genetics and trying to confirm that they were actually chlorophytes and haptophytes, but also looking at the phytoplankton and prokaryotes at different depth layers, because you can tell that something changed during these years over here. And it will be very interesting to target and see if the community changed during these years in relation to non-marine heatwave years. And really constrain the POC production export before, during, and after marine heat waves, and then tie with this phytoplankton and prokaryotic composition. That said, thank you. If you have any questions related to what I talked today, please send me an email. And if that's said, I'm done.
Thank you so much, Mariana. It's very impressive work. We are a little bit behind of time. We will move to a very little bit short discussion panel. Chenny, are you going to?
Yes, so I think that, sorry for those of you that have to leave now, but for people that can stay, let's try to just maybe have a 10 minutes discussion. And I know that there's been some chatter in the chat about QC that I think maybe would be worth at least saying a point on it, because a number of people were kind of making this point that for these global scale analyses, it does kind of require that there be consistent calibration being applied to all the floats. And I know at least for the GO-BGC and the SOCOM floats specifically, there's, you know, a lot of documentation of that, and many of the papers that were linked in the chat, you know, you can go to them to see the exact procedures. But maybe Yui or someone could make a comment about, you know, the GO-BGC protocol compared to floats that are being put out by other groups that are not, you know, affiliated, and how we can ensure that there is consistency in the data set globally, like including all floats, and not just the GO-BGC or the SOCOM floats. So I don't know, Yui, or if anyone wants to respond to that from the GO-BGC side, perhaps.
Yeah, so there is a, you know, there are very, you know, like documented protocols on how to correct the BGC-Argo data, and I believe there was a link that was posted to that, in the chat, you know, so those are well documented. The goal is that the data centers around the world will do the QA and the QC, and then those will be reflected in the flags, you know, and then we are working towards that goal. And then so all GO-BGC and SOCOM floats have gone through these QA/QC procedures, and, you know, certain parameters such as oxygen is getting better, and then we're working on pH and nitrate to kind of get the global data set to a more, you know, like uniformly QA/QC'd. So I wouldn't, the goal is to have the, you know, the DACs do it. So I'm not sure if it's a feasible solution. If individual groups have already done it, just to post their data might not be the most effective, I would say right now. I know that it's a little, it might be a little frustrating because the different data sets are in different statuses, but, you know, we're working very hard. The international community is working really hard to get that up and running. So I would say, you know, like still the point of contact, point to download the data are the SPROF files on the Argo GDAC files, and then just pay attention to the QF flags very, you know, closely. And then if there is a QF flag, you know, it says it's been QA/QC, then it has gone through those documented procedures.
Great, thank you. I just thought that was maybe worth saying.
Yeah, Jenny, this is Tanya Maurer. I'll just add to that too. I think Tom brought up a good point, in particular related to the chlorophyll A. You know, different parameters are sort of in different stages of development in terms of the delayed mode quality control procedures that are getting applied across the global data set. So oxygen, pH, nitrate are sort of somewhat more developed in terms of what's getting done to the data systemically across the whole array. As Yui mentioned, yes, looking in particular at the flags and the data mode is going to help the users, but we're still sort of in the early stages of developing procedures for each parameter. And we invite, researchers who've invested a lot of time and QA, QC or adjusting data and think that they have, insights, can provide insights for new methods and stuff. We always invite you and encourage you to reach out to data managers and attend the ADMT meetings. So yeah, we're really striving towards a unified system, and the ADMT framework is there to kind of vet these procedures as they're getting explored, so that they can be applied globally across stacks.
Great, thank you so much. And so now if anyone wants to, you can either raise your hand if you'd like to ask your question for any of the speakers, or sort of make any general comments about using the float data to kind of look at these questions of production and bloom phenology. I guess to start off, one question from the chat was about, so the fluorescence to chlorophyll ratio can vary, regionally. So whether that was accounted for at all in the classification method that you use, Nick, or if you just used, the same gain correction everywhere for the chlorophyll.
Yeah, I really just used the data as was being available. I didn't go back through and apply any additional corrections of my own.
Okay, thank you. So Ken, I see you have your hand raised if you want to unmute yourself and ask your question.
Thanks, Channing. Yeah, this is actually a question for Nicholas Bock. You were focused on the upper ocean chlorophyll, but I noticed in your Arabian Sea plot, you very nicely capture the backscatter in the oxygen minimum zone. And I know that's a thing. That's probably the bacteria doing the nitrate reduction, whatever, but I wonder if you had anything to say about that. There's a lot of interest in OMZs and the bacteria doing work there.
Yeah, no, and so we didn't really do anything to look at that in more detail. I think you're right. That was the same conclusion that we reached. The peaks, the sort of like mesopelagic peaks and backscatter measurements were in response to the populations of denitrifying bacteria, but we didn't do anything to try to quantify relationships between, particle stocks and magnitude of the mesopelagic PVP maximum or anything like that. Although that is something that I'm interested in working more on.
Another question for Nick that I was wondering is, have you ever tried applying the same classification method to just the surface chlorophyll values from the floats to kind of get some sense of whether the, surface values versus the integrated would give you, I would assume different classifications, since many of them are based off of DCM structure or depth, and that might help gain insight into the comparison of these bioregions to ones that are derived purely from surface chlorophyll?
Yeah, no, thanks for that question. We did try doing the, I'm trying to remember what depth range we used. I think it was maybe like the first 5 meters or so, to try to get a sense of how much extra information all the subsurface variability provides, and the method just doesn't work very well. The SVD results capture a relatively small proportion of the overall variance in the surface data compared to when we use like the full 1,000 meters from each float. So what we ended up doing was comparing the results to the example that I showed from Barenfeld et al. 2005 with the concept of these chlorophyll variance bins, to get saying, okay, well, this represents, these are the biomes that you might expect or you might find, based on the surface values alone. And then we compared those, sort of used a confusion matrix to show where our biome classifications overlapped and where there was more, where our results converged with the Barenfeld et al. 2005 results, and where there's more overlap. And what we found was that regions with really high chlorophyll concentrations or really, really low chlorophyll concentrations, the subsurface values didn't necessarily add as much. because it represents the polar regions and the oligotrophic regions respectively. But regions where there's regions characterized by intermediate surface chlorophyll concentrations, there's just a huge amount of overlap. Maybe the variance bin 3, the one that was right in the middle, captured the Southern Ocean, the Mediterranean, huge areas of the Indian Ocean, etc. And so that's really where our results provided more information than what you might be able to expect based on surface values alone.
Thanks. So we have a question from Allison Gray for any of the speakers, maybe Mari or Marin, since they haven't had a chance to respond to anything yet. But the question is, how do you think your results might evolve when the BGC-Argo array achieves a more uniform global coverage?
Yeah, I can go for one part of this one. So regarding the DCM, when I finished my study, we still lack of observation within the subequatorial regions, like Pacific and South Atlantic region. And I feel like these are really interesting because it's really region where we have a permanent high the biomass maximum. So it will be interesting to really map this distribution and also look at the seasonality. So for the DCM studies, and I think also related to Nicolas Bock studies, it's to try to have also this transition zone between really oligotrophic area and more mesotrophic, and I think more global observation will allow to better characterize the transition from one zone to the other.
Yes, and I think the more floats we have and the more data we have throughout the years, the more we can identify those marine heat wave events. So as we could see, there are a lot of marine heat waves everywhere. And in some places, there is not really enough data to capture those marine heat waves. There was a very interesting one that happened in the South Pacific Ocean, was a little bit north of the SOCOM floats. So we didn't really have data to look at, but that region is very interesting. So the more we have the floats there, the more we can capture these features. And we never know when these marine heat waves are gonna hit. So we always need to have the floats there.
Great. Well, thank you. And so perhaps on that note, we maybe should end here since we are a bit over already, but thank you again to our speakers. Thank you to all of you for coming. And I'm sure that any of the speakers would be happy to follow up via email if anyone has lingering questions that we didn't have time to address. But Eben, did you have anything else you wanted to add, or Yui, or anyone?
No, just thanks again for all the speaker and audience coming in.
Great. Well, we will reconvene, I guess, next quarter. These are going to be quarterly webinars, so sometime in the fall we will be having our next one. So thank you all.
Thank you for organizing and everything.
Thank you so much.
Thank you.